As we all know, we are surrounded by microorganisms which causes diseases. In the air, in our skin, in our food, pretty much everywhere. So why don't we get sick all the time? Have you ever wondered what causes allergy and what's the deal with vaccines? How do they work? All of these questions can be answered on understanding how immune system works. Good morning, and we are the Group 3 from R2A, and we will be discussing the specific host defense mechanism, an introduction to immunology. This will tackle the immunology, immune system, immunity, cells of the immune system, humoral immunity, cell-mediated immunity, hypersensitivity and hyposensitivity reaction, autoimmune diseases, immunosuppression, and the immunology laboratory. Immunology is the scientific study of the immune system and immune responses. Scientists who study various aspects of the immune system are called immunologists. The immune system is considered to be a specific host defense mechanism and the third line of defense. It is considered a specific host defense mechanism because it springs into action to defend against a specific pathogen or other foreign object that has gained entrance to the body. The origin of the immunology. Edward Jenner's smallpox vaccine in 1976 or Louis Pasteur vaccine against anthrax, cholera, and rabies in the late 1800s as the origin of the science of immunology. Pasteur is the first to use the term immune and immunity, but they don't know how the vaccines work. In 1890, immunology or the study of immunity started when Emil Bering and Kitasato Shibasaburo discovered antibodies while developing a diphtheria antitoxin. Also, Elie Michnikov discovered phagocytes and introduced the cellular theory of immunity. By 1910, the main elements of clinical immunology, the allergy, autoimmunity, and transplantation immunity, had been described. Major advances in immunology started in 1950s when the focus shifted from serology, or the investigating antigens and antibodies in serum, to cells. The discovery of lymphocytes marked the dawn of new era. The focus on immune cells and the rise of molecular biology concepts and methods were the two major effects on immunology since its inception. The first immunologic methods were devised to diagnose infectious disorders, which is where medical laboratory immunology originated. The key to understanding immunology. An understanding of immunology boils down to an understanding of two terms: antigens and antibodies. For the moment, think of antigens as molecules, usually proteins, that stimulates a person's immune system to produce antibodies. Think of antibodies as protein molecules that a person's immune system produces in response to antigens. Later in the chapter, antigens and antibodies will be discussed in more detail.
Today we are going to discuss regarding primary functions of the immune system. Firstly, the immune system consists of organs, cells, and substances that work together to combat illnesses, or what are known microbes. White blood cells, antibodies, the complement system, the lymphatic, and the spleen, the thymus, and the bone marrow are the key components of the immune system. Now, these are the immune system components that actively combat infection. Memory cells are white blood cells, and they're also called, B and T lymphocytes that keep track of every germ the immune system has ever fought. Now, if the microbe enters the body again, it will be recognized and destroyed promptly before it can proliferate and make you sick. Now, next is the major arms of the immune system. Now, on what we call the epidermis in the body tissues and in physiological fluids such as blood, the immune system, it fights the bacteria and other objects. The innate or what we call generic immune system and the adaptive or specific immune system make up the immune system. These two systems collaborate closely and perform various functions. Now, if the innate immune system fails to eradicate germs, the adaptive immune system takes over. And how? It targets the exact type of germ that is causing the sickness. So in order for For the system to do so, however, it must first detect the germ. Now, this implies it takes longer to respond than the innate immune system, but it is more accurate once it does. It also has the benefit of being able to remember or recognize pathogens, allowing the adaptive immune system to respond faster the next time a known germ is met. This memory is also the reason why some illnesses can only be contracted once in a lifetime, since your body becomes immune after that. So, one good example is the chicken pox. Once you had it, your immune system recognizes or having the memory of the chicken pox, and after that, then you won't be able to acquire the disease anymore for a lifetime. Next is immunity. Immunity can be defined as a complex biological system endowed with the capacity to recognize and tolerate whatever belongs to the cell. And to recognize and reject what is foreign or non-self is the capability of multicellular organisms to resist harmful microorganisms. Immunity involves both specific and non-specific components. The non-specific components act as barriers of eliminators of a wide range of pathogens irrespective of their antigenic makeup. And acquired immunity. Now, acquired immunity that arises when a person's immune system reacts to a foreign chemical or microbe, or when a person acquires antibodies from a different source. Adaptive and passive acquired immunity are the two forms of acquired immunity. Let's say when a person is infected with or vaccinated against a microbe, they develop adaptive immunity. So it's like the vaccination for COVID-19 nowadays. Now, the body responds with an immune response that can help prevent subsequent infections with the microbe. When a person obtains antibodies against a disease or toxin, rather than creating themselves, he or she is said to have passive immunity.
Vaccination. It has been known since the time of ancient Greeks that persons who have recovered from infectious diseases like plague, smallpox, and yellow fever rarely get them again. The use of vaccinations to prevent diseases may date back to the 11th century, when the Chinese employed powder made from dried smallpox scabs to immunize people either by ingesting or injecting it into their bodies. This method was known as the Chinese technique. Edward Jenner, a British physician, was one of the individuals who inoculated this way. Jenner later looked into the prevalent idea that milkmaids who often had smooth, thin skin never got smallpox. The milkmaids were protected by smallpox by a vaccine that left no scars. That is where Jenner created the smallpox vaccine from cowpox lesion material. People who received Jenner's vaccinations were immune to smallpox. For the cow, Jenner is credited by being the first to publish the effective outcomes of vaccinations in 1798. Louis Pasteur developed a successful vaccinations to prevent cholera in chickens, anthrax in sheep, cattle, and rabies in canines in the last 1800s. And Pasteur was the one who initially used the phrases vaccinations and vaccine. It is defined as material that can artificially induce immune to an infectious disease, usually after injection or in some cases ingestion of some materials. It also exposes a person to a harmless version of pathogen to stimulate the person's immune system to produce protective antibodies and memory cells. An ideal vaccine is one that contains enough antigenic determinant to stimulate the immune system to produce protective antibodies, contains antigenic determinants from all the strains of the pathogen that cause the disease, has no or few side effects, does not cause diseases in the vaccinated persons. Types of available vaccines. Various materials are used in vaccines. Most vaccines are made from living or dead pathogens or from certain toxins they produce. The use of such vaccines illustrates a very important and practical application of the principle of microbiology and immunology. Children should receive the following vaccines between birth and entry to school: hepatitis B vaccine, rotavirus vaccine, diphtheria toxoid tetanus vaccine, inactivated poliovirus vaccine, MMR vaccines, varicella vaccine, influenza vaccine, pneumococcal vaccine, hepatitis A vaccine, meningococcal vaccine. Types of available vaccines. Attenuated vaccines. The process of weakening pathogen is called attenuation, and the vaccines are referred to as attenuated vaccines. Attenuated viral vaccines are the examples of adenovirus, chickenpox, measles, mumps, German measles, polio, rotavirus, smallpox, and yellow fever. Inactivated vaccines made from pathogens that have been killed by heat or chemicals called inactivated vaccines. It can be produced faster and more easily, but they have less effective than live vaccines. This is because the antigens on the dead cells are usually less effective and produce a shorter period of immunity. Subunit vaccines. This is the ones that uses antigenic or antibody stimulating portions of a pathogen rather than using the whole pathogen. Vaccines have been made by conjugating bacterial capsular antigens, which by themselves are not very antigenic, to molecules that stimulates the immune to produce antibodies against the less antigenic capsular antigens. Toxoid vaccines is an exotoxin that has been inactivated or made non-toxic by heat or chemicals. Toxoids can be injected safely to stimulate the production of antibodies that are capable of neutralizing the exotoxins of pathogens. Vaccines or gene vaccines are only experimental. A particular gene from a pathogen is inserted into plasmids, and then the plasmids are then injected into the skin or tissue. Autogenous vaccines. One that has been prepared from bacteria isolated from a localized infection such as the staphylococcal boil. The pathogens are killed and then injected into the same person to induce productions of more antibodies. How vaccines work. Vaccines cause the immune system of a recipient to create protective antibodies. The protective antibodies and/or memory cells produced in response to the vaccine then remain in the recipient's body to fight a specific infection if it ever enters the recipient's body a point in the future. Passive acquired immunity. In passive acquired immunity, a person receives antibody rather than producing them. This can occur naturally or in an artificial manner. In natural passive acquired immunity, a fetus receiving maternal antibodies in utero and an infant receiving maternal antibodies in the colostrum are examples of natural passive acquired immunity. Artificial passive acquired immunity is accomplished by transferring antibodies from an immune person to a susceptible person. After a patient has been exposed to a disease, the length of the incubation period usually does not allow sufficient time for post-exposure vaccinations to be an effective preventive measure. This is because a span is about 2 weeks is needed before sufficient antibodies are formed to protect the exposed person. Receiving a shot of gamma globulin is an example of artificial passive acquired immunity. The immune system. As stated earlier, the immune system involves very complex interactions among many different types of cells and cellular secretions. The major cell types that participate in immune responses are: number 1, T lymphocytes or T cells; number 2, B lymphocytes or B cells; number 3, NK cells or category of lymphocytes; number 4, macrophages. The cells involved in immune responses originate in the bone marrow, from which most blood cells develop. 3 lines of lymphocytes, B lymphocytes or B cells, T lymphocytes or T cells, and NK cells are derived from lymphoid stem cells of the bone marrow. There are 2 major categories of T cells: the helper T cells and the cytotoxic T cells. Helper T cells are also known as the T helper cells, TH cells, and CD cells. The term CD4 cells refers to the fact that these cells possesses on their surface an antigen designated as CD4. The primary function of T helper cells is secretion of cytokines. TH1 cells and the TH2 cells are subcategories of the helper T cells. cytokines secreted by Th1 cells to support cell-mediated immune responses involving macrophages, cytotoxic T cells, and NK cells. Cytokines secreted by Th2 cells support humoral immune responses by inducing B cells activation and the differentiation of activated B cells into plasma cells. Cytotoxic T cells are also known as T cytotoxic cells, TC cells, or CD8 cells. The term CD8 cells refers to the fact that these cells possesses on their surface an antigen designated as CD8. The primary function of cytotoxic T cells is destroy virally infected host cells, foreign cells, and tumor cells. Humoral immunity. In humoral immunity, special glycoproteins are called antibodies, are produced by B cells in response to antigens. In many cases, these antibodies are capable of recognizing, binding to, and inactivating or destroying specific pathogens. A bacterial cell could be described as a mosaic of antigenic determinants. The important point is that, in most cases, antigen must be foreign materials that the human body does not recognize as a self-antigens. Certainly, all invading microbes fall into this category. Some small molecules called haptens may act as antigen only if they are coupled with a large carrier molecule such as a protein. Individual molecules that stimulate the production of antibodies are referred to as antigenic determinants or epitopes.
Processing of antigens in the body. For antibodies to be produced within the body, a complex series of events must occur. It is known that macrophages, T cells, and B cells are often involved in a cooperative effort. Antigens are classified into two: the T-dependent cells and the T-independent cells. T-dependent antigens. The majority of antigens are referred to as T-dependent antigens because T cells, specifically Th cells, are involved in their processing. The processing of T-dependent antigens also involves macrophages and B cells. T-independent antigens, the processing of which only requires only B cells. T-independent antigens are large polymeric molecules, usually polysaccharides containing repeating antigenic determinants. Examples include the lipopolysaccharide or LPS found in the cell walls of gram-negative bacteria, bacterial flagella, and bacterial capsule. The processing of other category of antigen T-dependent or T-independent result in B cells developing into plasma cells that are capable of secreting antibodies. T-dependent mechanism. Step 1. Processing of T-independent antigens is initiated when an appropriate B cell makes physical contact with the free antigenic determinant. Step 2. The activated B cells undergoes extensive cell division, producing a clone of identical B cell. Step 3. Some of the members of the newly formed clone mature into antibody-producing plasma cells, whereas others become more memory cells. T-independent mechanism. Step 1. After invasion of the body, an antigen is ingested and digested by a macrophage. Step 2. Within the macrophage, antigenic determinants of the bacterial cell attach to the molecule called major histocompatibility complex, or MHC molecule. Step 3. The combined AT-MHC molecules are then displayed on the surface of the macrophage. The macrophage is referred to as an antigen-presenting cell or APC. Step 4. A Th cell attaches to one of the AP-MHC molecules, divides and sends out chemical signals. Step 5. When the chemical signals reach a B cell that is capable of recognizing that particular signal, the activated B cells divides, producing a clone of identical B cells. Step 6. Antibodies are expelled rapidly for several days until the plasma cell dies. Each plasma cell makes only one type of antibody, one that will bind with the antigenic determinant that activated the B cell and stimulated production of the antibody. A closer look at antibodies. Antibodies are proteins produced by lymphocytes in response to the presence of an antigen. A bacterial cell has a numerous antigenic determinants on its cell membrane, cell wall, capsule, and flagella that stimulate the production of many different antibodies. Usually, an antibody is
specific in that it will recognize and bind to only antigenic determinant that is stimulated by its production. All antibodies are in a category of proteins called immunoglobulins, globular glycoproteins in the blood that participate in immune reactions. The term antibodies is used to refer to immunoglobulins with particular specificity for an antigen. The antibody structure. As previously mentioned, antibodies belong to a category of glycoproteins called immunoglobulins. All antibodies are immunoglobulins, but not all immunoglobulins are antibodies. The basic structure of an immunoglobulin molecule resembles to the letter Y. It consists of two identical light polypeptide chains, two identical heavy polypeptide chains, two antigen binding sites, and an Fc region. In this basic form, the molecule is referred to as a monomer. The immunoglobulin classes. Studies of gamma globulin component of human blood have revealed that 5 classes of immunoglobulins exist: the IgA, IgD, IgE, IgG, and IgM. IgA, it is the predominant immunoglobulin class in saliva, tears, seminal fluid, colostrum, breast milk, and mucous secretions of the nose, lungs, and gastrointestinal tract. IgD, it is found in large quantities on the surface of B cells. Its function is unknown, but it's possible that IgD molecules on the B cell's surface serve as an antigen receptors and determine which antigen that particular B cell is able to respond to. IgE. In atopic individuals, IgE is produced in response to allergens found on the surfaces of basophils and mast cells. It plays a major role in allergic responses. IgG, the only class of immunoglobulin that can cross the placenta. Maternal IgG antibodies that cross the placenta help protect the newborn during its first months of life. Antigen-bound IgG can bind and activate complement, and that process is known as complement fixation. IgG molecules can bind to a wide range of cellular receptors to promote phagocytosis and antibody-dependent cytotoxicity. IgM. IgM antibodies are the first antibodies formed in the primary response to antigens including pathogens. Although IgG antibodies later become the most prevalent class, IgM antibodies are relatively short-lived, remaining in the bloodstream for only a few months. Because of its large size, IgM does not cross the placenta.
How antibodies protect us from pathogens and infectious diseases. Once they are produced, antibodies are very specific. Usually, a given antibody can only recognize and bind to the antigenic determinants that stimulate its production. Example, a person is infected with an encapsulated bacterium. Recall that bacterial capsules serve an antiphagocytic function, meaning that phagocytic white blood cells are unavailable to phagocytize encapsulated bacteria. The reason for this is that the phagocytes have no receptors on their surface that recognize the polysaccharide molecules. If the phagocyte is unavailable to attach to the encapsulated bacterium, it is unavailable to phagocytize it. That person's immune system responds by producing antibodies against the capsular polysaccharide molecules. The antibodies attach to the capsule. This makes it possible for the phagocytes to bind the encapsulated bacteria. Why? Because the phagocytes have receptors on their surface that can recognize and bind to the antibody molecules. Among other functions, antibodies can neutralize toxins, prevent attachment of pathogens to host cells, and promote phagocytosis. Antigen-antibody interaction and their effect. Interaction, prevention of attachment. Effect, a pathogen coated with antibody is prevented from attaching to a cell. Clumping of antigen. Antibodies link antigen together, forming a cluster that phagocyte can ingest. Neutralization of toxin. Antibodies bind to toxin molecules to prevent them from damaging cells. Help with phagocytosis. Phagocytes can attach more easily to antigens that are coated with antibody. Activation of complement. When complement attached to the antibody on a cell surface, a series of reactions begins that activates complement to destroy cells. Activation of NK cells. NK cells respond to antibody adhering to cell surface and attack the cell. Monoclonal antibodies. Purified antibodies that are directed against specific antigens have been produced in laboratories by innovative technique. in which a single plasma cell that produces only one specific type of antibody is fused with rapidly dividing tumor cells. The new long-lived antibody-producing cell is called hybridoma. These hybridomas are capable of producing large amounts of specific antibodies called monoclonal antibodies. Cell-mediated immunity. Cell-mediated immune responses involve many types of cells and cytokines. Antibodies are infrequently, if ever, involved. Antibodies are unavailable to enter cells, including cells containing intracellular pathogens. Typical cell-mediated cytotoxic response would involve the following steps. Step 1. A macrophage engulf and partially digest a pathogen. Fragments, antigenic determinants of the pathogen are then displayed on the surface of macrophages. The macrophages act as an antigen-presenting cell. Step 2. A Th cell binds to one of antigenic determinants being displayed on the macrophage surface. The Th cells produce cytokines, which reach an effector cells of the immune system, for example, T cells or NK cells. Step 3. The effector cell binds to a target cell, for example, pathogen-infected host cell displaying the same antigenic determinants on its surface. Step 4. Vesicular content of the effector cells are discharged. This includes perforin and other proteins and enzymes, which literally punch holes in the target cell membrane. Other cytokines released by effector cells are tumor necrosis factor and NK cytotoxic factor. Step 5. Toxins produced by the effector cells enter the target cell, causing disruption of DNA and organelles. The target cells dies. Natural killer cells. Cytotoxic T cells and NK cells kill foreign cells. Host cells infected with viruses or bacteria and tumor cells. NK cells are in a subpopulation of lymphocytes called large granular lymphocytes. Although they morphologically resemble lymphocyte, NK cells lack typically T or B cell surface markers. They also differ from T and B cells in other ways. For example, they do not proliferate in response to antigen and appear not to be involved in antigen-specific recognition. As name implies, NK cells kill target cells, including foreign cells, host cells infected with virus or bacteria, and tumor cells. Although NK cell activity is not dependent on antibodies, NK cells have receptors on the surface of the Fc region of IgG antibodies. These receptors enable the cell to attach to and kill antibody-coated target cells. This is known as antibody-dependent cellular cytotoxicity. Once attached to an antibody-coated target cell, the NK cell inserts a molecule called perforin into the cell membrane of the target cell, creating an opening through which cytotoxic granules called enzymes are injected. Although firm evidence is lacking for an immune surveillance system within our bodies that monitors for and destroys malignant cells, NK cells may participate in such a system.
Hypersensitivity and hypersensitivity reactions. It refers to undesirable reactions produced by the normal immune system, including allergies and autoimmunity. They are usually referred to as an overreaction of the immune system in an attempt to protect the person, causing irritation or damage to certain cells and tissues in the body. And these reactions may be damaging and uncomfortable. There are several types of hypersensitivity reactions. Some types involve antibodies, whereas others do not. All types depend on the presence of antigen and T cells that are sensitized to that antigen. Hypersensitivity reactions are divided into two general categories, the immediate type and delayed type. The immediate type hypersensitivity reactions occur from within a few minutes to 24 hours after contact with a particular antigen. There are three categories of immediate type hypersensitivity reactions, referred to as type 1, type 2, and type 3. A delayed type hypersensitivity or DTH reaction usually takes more than 24 hours to manifest itself. DTH reactions are also known as type 4 and cell-mediated reactions. The immediate type of hypersensitivity reactions occur from within a few minutes to 24 hours after contact within a particular antigen. So that's type 1, which is the anaphylactic reactions or the allergic reactions. The type 2 hypersensitivity reactions or the cytotoxic reaction involve damage to or death of body cells. The type 3 hypersensitivity reactions is the immune complex reaction. Damage to tissues and organs is initiated by antigen antibody complexes. So the DTH reaction that usually takes 24 to 48 hours or longer to manifest themselves is the type 4, or also known as the cell-mediated reactions, where the antibodies plays only a minor role, and if any, an example is a positive TB skin test. So the type 1 hypersensitivity reaction, are also known as anaphylactic reactions, include classic allergic responses such as hay fever symptoms, asthma, hives, and gastrointestinal symptoms that result from food allergies or allergic responses to insect stings and drugs, and anaphylactic shock. So the type 2 hypersensitivity reaction involves antibody-mediated destruction of cells. It is also known as the cytotoxic reaction. And in this hypersensitivity reaction, a specific antibody, the IgG or IgM, binds to the cell surface antigen and destroys the cells. And if the cell is a microorganism, killing of the cell is beneficial to the host. However, in type 2, the cells have their own RBC. So the type 3 hypersensitivity reaction are also known as immune complex reactions. The examples of type 3 are the serum sickness and certain autoimmune diseases like systemic lupus erythematosus or SLE and rheumatoid arthritis. These reactions involve IgG or IgM antibodies, complement, and neutrophils. If immune complexes are not removed from blood, they accumulate on the wall of blood vessels and on tissue, where filtration of blood and plasma occurs, such as glomerular membrane, synovial membrane of joints. The position of immune complexes initiates reaction, resulting in damage of surrounding tissue and causes inflammation. So lastly is the type 4 hypersensitivity reaction, are also known as the delete type, or cell-mediated immune reaction. It typically takes 24 to 48 hours to manifest themselves. They occur in tuberculin and fungal skin tests, contact dermatitis, and transplantation rejection. The type 1, the IgE-mediated, with a quick onset after exposure of the allergic, like bee stings, latex, and certain medications like penicillin. So type 2 is the cytotoxic or antibodies mediated, like hemolytic reactions, good Asher syndrome, and hyperacute graft rejection. So the type 3 is the immune complex or the IgG and IgM mediated. So this is the hypersensitivity pneumonitis, systematic lupus erythematosus, and the serum sickness. The last one is the type 4, which is a delayed or cell-mediated, which is the chronic graft rejections, PPD test, latex and nickel, and poison ivy. Autoimmune diseases. An autoimmune disease results when a person's immune system no longer recognizes certain body tissues as self and attempts to destroy those tissues as if they were non-self or foreign. The autoimmune disease occurs when, instead of attacking bacteria, viruses, or other sources of infection, the immune system attacks healthy organs and tissues. The cause of autoimmune disease is this may occur with certain tissues that are not exposed to the immune system during fetal development so that they are not recognized as self. It's not known why autoimmune diseases develop, although most people who have an autoimmune disease have a genetic predisposition. So an environmental factor such as an infection, stress, medication, diet, or even ultraviolet radiation then triggers the symptoms of the autoimmune disease. Autoimmune diseases can be classified as organ-specific and non-organ specific. Examples of organ-specific autoimmune diseases are Hashimoto, thyroiditis, the Graves' disease, and the primary myxedema, thyrotoxicosis, all three of which affect the thyroid. So the non-organ specific autoimmune diseases involve the skin, kidneys, joints, and muscles. Immunosuppression. If a person's immune system is functioning properly, he or she is said to be an immunocompetent person. If a person's immune system is not functioning properly, he or she is said to be immunosuppressed, immunodepressed, or immunocompromised. The most common cause of immunodeficiency worldwide is malnutrition. It is a suppression of the immune system and its ability to fight infection. Immunosuppression may result from certain diseases such as AIDS or lymphoma, or from certain drugs, such as some of those used to treat cancer. So immunosuppression may also be deliberately induced with drugs, as in preparation for bone marrow or other organ transplantation to prevent the rejection of a transplant, or also known as immunodepression. So the immunodeficiencies may be either inherited or acquired. So the acquired immunodeficiencies may be caused by drugs, for example, cancer chemotherapeutic agents and drugs given to transplant patients, irradiation, or certain infectious diseases like HIV infection. So the inherited immunodeficiency diseases can be a result of deficiencies in antibody production, complement activity, phagocytic function, or NK cell function. The several inherited immunodeficiency diseases are chronic granulomatous diseases, and patients have deficiencies of B cells or T cells, or both, resulting in severe recurrent infections. So, a person lacking the ability to produce antibodies is said to have a gamma globulinemia, whereas a person who produces too few antibodies is said to have a hypogammaglobulinemia.
The Immunology Laboratory. There are 4 possible explanations for the presence of antibodies to a particular pathogen. Present infection, when the person is currently infected with the pathogen. Past infection, if the person was infected with the pathogen in the past and antibodies are still present in his or her body. Next is vaccination. The antibodies are a result of the person having been vaccinated against a particular pathogen at some time in the past. For example, a person's serum may contain antibodies against influenza viruses because he or she received a flu shot last year. Although we consider antibodies to be very specific, there are instances where antibodies can react with epitopes that are molecularly similar to but not identical to the epitope that stimulated production of these antibodies. This is known as cross-reactivity for a cross-reaction. Fortunately, in most IDPs, cross-reactivity is not a common event. Next, we will be discussing about blood typing. Agglutination tests are used in the blood bank to learn a person's blood type, which is determined by the 2 types of antigen present to the surface of his or her RBCs. 3 reagents are used for the ABO and Rh typing antigen. A antiserum, or a serum containing antibodies against A antigen. Anti-B antiserum, or the A serum containing antibodies against B antigen. Anti-Rh antiserum, or A serum containing antibodies against Rh antigen H. Example number 1. A person is said to be A positive if his/her RBCs have A antigen and Rh antigen on their surface but lack B antigen. Second example, a person is said to be O negative if his or her RBCs lack A antigen, B antigen, and Rh antigen. It is important to remember that in addition to the antigens that determine a person's ABO blood type and Rh status, the surface of RBCs contains many other antigens. Immunization is one of the best ways we can protect ourselves, our children, and our future generations from infectious diseases. In other words, if you vaccinate, you help wipe out disease that could spread now and into the future. With that all being said, I hope that you learned something today and use it in our future endeavors. Thank you and good day.